Hi everybody. Today we shall discuss some of the questions from the chapter Microbes in Human Welfare. In NEET, usually one, rarely two questions are asked from this chapter. So this is a relatively easy chapter and mainly dependent on the remembrance. So that's why carefully you have to read the chapter prior to the exam. One day before the exam, once again you go through all the important points that have to be remembered in this particular chapter. Let us see the questions that can be asked from this particular chapter, one after the other. Which among the following is not used for human welfare? Which is not used for human welfare? So all the four options that are given are microbes. Agrobacterium tumefaciens, it is used for human welfare. Its plasmid is used as gene cloning vector in plant genetic engineering. It is used for human welfare. Bacillus thuringiensis used for human welfare. Bacillus thuringiensis is used as biological control to eradicate insect pests that damage the crop plants. E. coli. E. coli is also used for human welfare because it is the main microbial host that is used in genetic engineering. Right? Next one, prions. So prions are not used for human welfare because prions cause diseases in human beings. They are not used so far, they are not used for human welfare. So prions must be the correct option. Let us see the answer. Fourth one. Here is the explanation. Agrobacterium tumefaciens is used in plant genetic engineering to obtain genetically modified plants. Bacillus thuringiensis is used as biological control to eradicate insect pests of crops. E. coli is used as heterologous host in rDNA technology. Prions are not used in human welfare as they neither serve as vectors in genetic engineering nor used in biological control and other beneficial programs to mankind. Coming to the next question. Statements. Given below are two statements. So statement 1. Adenovirus causes respiratory infections in man. Absolutely true. It causes respiratory infections in man. Statement 1 is correct. And adenovirus is rod-shaped virus. No, it is polyhedral. Or it has got icosahedron symmetry or the polyhedral virus. Statement 2 is wrong. Statement 1 is correct, but statement 2 is incorrect. First option must be the correct one. Let us see the answer. First one. Here is the explanation. Adenovirus causes respiratory infections in man. It is not rod-shaped, it is polyhedral or icosahedron symmetry it has got. Polyhedral in shape. Coming to the next question. Propionibacterium shermani is used for the production of. It is a direct question that is given in the NCERT book. Propionibacterium shermani is used for the production of Swiss cheese. Roquefort cheese is the cheese ripened by using Penicillium roqueforti. Camembert cheese is ripened by using the Penicillium camemberti. Swiss cheese is produced with large holes by using Propionibacterium shermani. Not Indian cheese. No. So Indian cheese is not the correct answer. Swiss cheese is the correct answer. Second option must be the correct one. Let us see the answer. Second one. Roquefort cheese is ripened by using a fungus Penicillium roquefort. Camembert cheese is ripened by using Penicillium camemberti fungus. Swiss cheese with larger holes is made by fermenting with Propionibacterium shermani. No microbe is used in making Indian cheese. So whatever microbes that are present in the air flora or air microbial flora, they are involved in Indian cheese making. Coming to the next question, fourth one. Assertion reason type. The nutritional quality of a food is improved during curdling of milk by lab. Absolutely true. Nutritional quality is increased. So it is due to the formation of vitamin B12 by the lab bacteria. So vitamin B12 as such is not present in the milk. During the growth of the lactobacilli or lactic acid bacteria, during curdling of the milk, they also prepare the vitamin B12 and release into the curd. So assertion is correct, reason is correct, reason is clearly explaining the assertion. So option 4 must be the correct one. Let us see the answer. 4. During curdling of milk, not only the lactic acid bacteria involved in formation of curd from milk, but also improve the nutritional value of the curd by increasing the content of vitamin B12. Coming to the next question. Which microbe is used for the production of fermented beverages? Fermented beverages. Fermented beverages only yeasts are used. That is Saccharomyces. Lactobacillus. No. It is used for the production of curd, curdling of milk, or lactic acid production also. Iconostarch. It is the fermentation of the dosa and idli batter. Penicillium. Penicillium is used for antibiotic production, not used for fermented beverages. Right? Penicillium species are also used for ripening of the cheese. Saccharomyces, yes, this is the fungus, belongs to Ascomyces, which is unicellular and used in the production of fermented beverages by fermenting various substrates like malt, red fruit juices, etc. So answer must be the fourth one. Let us see the answer. Fourth one. Saccharomyces cerevisiae or yeast is used for the production of fermented beverages. Lactobacillus is used for curdling of milk. Penicillium is used for the ripening of certain cheeses. Iconostarch species are involved in fermentation of certain foods like idli and dosa batter. Coming to the, this is matching type. Right? Discovery of which product earned Fleming the Nobel Prize? Penicillin. A4. So A4 is second and third options. So either second and third option must be the correct one. Produced industrially by using Aspergillus niger. That citric acid is produced. So B3. A4, B3. So second option must be the correct one. Produced in large scale by using Monascus purpureus. So which is produced in large scale? Statins are produced with 2. C2. Naturally, D should be 1. Produced in large scale by using Trichoderma polysporum. Trichoderma polysporum is used for the production of cyclosporin A. So it is used as immunosuppressive agent. Statins, so prevent the formation of cholesterol. Statins act as competitive inhibitors of the enzymes involved in cholesterol synthesis. So second option must be the correct one. Let us see the answer. Second one. Penicillin earned Fleming the Nobel Prize. Citric acid is produced by using Aspergillus niger. Statins are produced by using Monascus purpureus. Cyclosporin A is produced by using Trichoderma polysporum. Coming to the next question. Which among the following pair of alcoholic beverages are produced without the distillation of fermented broth? Without the distillation of fermented broth, what are produced? One is beer, another one is wine. Remaining brandy, whiskey, rum, gin, etc. They are all produced by distillation of fermented broth. Beer and brandy, beer is okay but not brandy. Whiskey and rum, both are found by distillation process. Beer and wine, this is correct, beer and wine. Rum and brandy, no. Third option must be the correct one. Let us see the answer. Third one. Whiskey, rum and brandy are produced by distillation of fermented broth. Beer and wine are not formed upon distillation of fermented broth. Without distillation, they are directly consumed. That is about the explanation. Coming to the next question, eighth question. Identify the correct statements out of the 5. Which are the correct ones that you have to identify? Streptokinase is the clot buster. Yes. It helps in dissolving the blood clots formed in the arteries. Right? Cyclosporin A is immunosuppressive agent. Yes. So streptokinase is produced by using streptococcus. Cyclosporin A is produced by using Trichoderma polysporum. Statins are competitive inhibitors of enzymes of cholesterol synthesis. This is also true. A, B, C are correct statements. Clostridium species is used for commercial production of butyric acid. Absolutely true. Clostridium bitylicum is used for the commercial production of butyric acid. Penicillin is antagonistic to Staphylococcus. Absolutely true. So while carrying out his experiments on Staphylococcus aureus, so Fleming discovered the synthesis of a chemical substance by a mold growing around Staphylococcus, that is the Penicillium notatum, that prevented the growth of Staphylococcus aureus. So, penicillin is antagonistic, opposite effect to Staphylococcus. This is also true. A, B, C, D, E, all the five statements are correct. So, fourth option must be the correct one. Let us see the answer. Fourth one. Streptokinase is used as clot buster to remove clots in blood vessels of patients undergone myocardial infarction. Cyclosporine A is used as immunosuppressive agent in organ transplant patients. Statins are competitive inhibitors of enzymes of cholesterol synthesis. Clostridium botulinum is used for butyric acid production. Penicillin is antagonistic to gram-positive bacteria like Staphylococcus aureus. Coming to the next question. Choose the incorrect statement. Ethanol is commercially produced by using Saccharomyces cerevisiae. Absolutely true. So, yeast is not only used for the formation of various beverages, for commercial production of alcohol also Saccharomyces cerevisiae is used. Right? Lipases are used in detergent formulations to remove oil stains. So, first statement is correct statement. Lipases are used in detergent formulation to remove oil stains. This is also correct. Pectinases and proteases are used as clarifying agents of bottled fruit juices. This is also correct. Fruit juices are clarified by using pectinases and proteases in juice industry. Fleming discovered penicillin while working with Streptococcus. No, he discovered penicillin while working with Staphylococcus, not Streptococcus. Fourth option is the incorrect one. Let us see the answer. Fourth one. Alexander Fleming discovered penicillin secreted by a mold, Penicillium notatum, while working with Staphylococcus aureus, not Streptococcus, which causes various infections related to skin and wounds in man. Coming to the next question. Statement 1, statement 2 type. Effluent and primary sludge are formed in primary settling tank of STP. Absolutely true. So, during physical treatment or primary treatment, the sewage is allowed to pass through the primary settling tank, allowed to pass into the primary settling tank. While allowing it to pass into the primary settling tank, sequential filtration is carried out. After that, in the primary settling tank, whatever the heavy objects are present like silt, pebbles and all that sand settled at the bottom of the tank, which is known as primary sludge. And the supernatant is referred as effluent. The supernatant of the sewage formed in primary settling tank is known as effluent. Supernatant of primary settling tank is called primary sludge. This is wrong. Primary sludge is the settled one, which consists of sand, silt, grit, etc. Right? So statement 2 is wrong. Statement 1 is correct. Statement 2 is wrong. Statement 1 is correct. Statement 2 is incorrect. First one is correct. Let us see the answer. First one. The sediment of primary settling tank is called primary sludge, and the supernatant with the dissolved and suspended organic matter is called effluent. Coming to the next one. Choose the incorrect statement. You have to choose the incorrect one. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. Absolutely true. During sewage treatment, actually the sewage is treated by using the heterotrophic microorganisms present in the sewage. They are exploited for the treatment. So this is correct statement. Both anaerobic bacteria and aerobic bacteria are involved in treating the sewage. This is also true. In aeration tank, aerobic bacteria are involved. In anaerobic sludge digester, to degrade the flux, there is involvement of anaerobic bacteria, particularly methanogenic bacteria. The second statement is also correct. Fungi are involved in treating the sewage in aeration tank. Yes, absolutely true. Fungi are involved. So both the aerobic fungi as well as bacteria are involved in treating the sewage in aeration tank, which decrease the amount of organic matter by consuming the oxygen. So gradually the organic content decreases, organic matter content decreases in the aeration tank, and population or the growth of aerobic bacteria and aerobic fungi increases. Flux are formed. Third option is also correct. Methanogens play a role in treating the sewage in aeration tank. No. Methanogens play a role in treating the sewage in anaerobic sludge digester. Anaerobic sludge digester, not in aeration tank. Aeration tank for sewage treatment, aerobic heterotrophic bacteria and fungi are used. So fourth option must be the incorrect one. Let us see the answer. Fourth one is the incorrect one. Methanogens play a role in treating the sewage, that is inactivated sludge, consisting of elementous fungi and aerobic bacteria in anaerobic sludge digester. Coming to the next question. In which of the following stages the BOD of sewage effluent is reduced? So, in which of the following stages the BOD of sewage of effluent is reduced? Primary treatment, no. It is physical treatment. Anaerobic sludge digester, no. In anaerobic sludge digester, the flux are degraded by methanogens or anaerobic microbes. Treatment in aeration tank, absolutely true. This is the correct one. Secondary settling tank where the flux are pumped, no. In secondary settling tank, after treating the sewage in aeration tank, that is passed into secondary settling tank where the flux are sedimented. The flux are sedimented. Right? That sediment is transferred to the anaerobic sludge digester. So, secondary treatment in aeration tank, the BOD of sewage effluent is reduced. Third option must be the correct one. Let us see the answer. Third one. BOD of sewage is reduced to permissible levels during the treatment in aeration tank. In primary treatment, floating debris is removed by sequential filtration and the silt and sand are removed by sedimentation. It is physical treatment. In anaerobic sludge digester, the flux are degraded by anaerobic bacteria and biogas is produced. In secondary settling tank, the flux are sedimented. Choose the correct sequence of events followed in treating the sewage in STPs. Correct sequence. Digestion of activated sludge. No, it is not the first one. It is digestion of activated sludge that occurs in anaerobic sludge digester. Digestion of dissolved and suspended organic matter in sewage. This is in aeration tank. So it comes prior to the A. B should come prior to the A. Formation of primary sludge. Formation of primary sludge is formed in primary settling tank. Removal of floating debris by sewage. Debris of sewage. That is during sequential filtration. B is first, next to C, next to B, next to A. BCBA. Second option must be the correct one. Let us see the answer. Second one. The various stages of sewage treatment in STPs and sequence is removal of floating debris of sewage by sequential filtration. It is a part of physical treatment. Next one. By sedimentation, the grit of sewage is removed. The sedimented grit is called primary sludge. In the primary settling tank, it is the sewage is allowed to sediment and the grit and everything, the sand, silt, etc., they are sedimented. So it is after filtration in physical treatment, not biological treatment. Next one. The organic matter of effluent is oxidized in aeration tank by exploiting the naturally present aerobes of sewage. Yes, this is done in aeration tank. Right? So the effluent of the primary settling tank is passed into the aeration tank where the organic matter of the sewage is considerably decreased by heterotrophic aerobic bacteria and fungi. B. Next one. Activated sludge consisting of aerobic filamentous fungi and bacteria is degraded in anaerobic sludge digester by exploiting the anaerobic methanogens present in sewage. Yes, this is the correct sequence. BCBA. Coming to the next question. Two statements are given. Statement 1. Ganga action plan and Amana action plan are related to building of STPs. Absolutely true. Biogas consists of only methane. This is wrong. Biogas consists of not only methane, some other gases like carbon dioxide, hydrogen sulfide, and molecular hydrogen are also liberated during biogas production. But main constituent is methane. Not only methane, main constituent is methane, but also other gases are present. Statement 1 is correct, statement 2 is incorrect. So, first option must be the correct one. First option. Ganga action plan and Yamuna action plan are related to building our STPs near cities to save the major rivers of our country from pollution. Yes. Biogas not only consists of methane but also hydrogen and carbon dioxide and even hydrogen sulfide or H2S. Coming to the next question. Choose the incorrect statement. Biogas plant has one inlet and two outlets. One inlet and two outlets. Absolutely true. This is correct statement. Not incorrect statement. We want incorrect statement. Inlet of biogas plant is connected to concrete tank at a lower level than its outlet. Absolutely true. If you look at the diagram that is given in the textbook, The inlet is connected at the base of the concrete tank, right? At the base of the concrete tank. The outlet is connected at upper level. To the floating tank, the outlet for biogas transport is connected. Floating cover is connected to the outlet where the spent slurry is collected. This is wrong. Floating cover is connected to the outlet where the spent slurry is collected. No. Floating cover is connected to the outlet through which the gas is supplied to the near houses. But the spent slurry, the outlet for spent slurry is connected to the concrete tank or cement tank or digester at the upper level. So third one is the incorrect one. The technology of biogas production was developed in India by IARI and KVIC, Khadi and Village Industries Commission, KVIC. IARI means Indian Agricultural Research Institute. So fourth option is correct statement, third option is incorrect statement, third one must be selected, the third one must be the answer. Okay. Floating cover is placed over the slurry or digester which is connected to a pipe to supply biogas to nearby houses but not to the outlet where the spent slurry is collected. Coming to the next question. Assertion reason type. So, dried spores of assertion is dried spores of Bacillus thuringiensis mixed with water is sprayed over brassicas and fruit trees. This is the textbook statement. The statement is correct. Brassicas and fruit trees are more vulnerable to harmful insect larvae that eat the plants. Absolutely true. So that is why sprayed over brassicas and fruit trees. Sprayed over. Because brassicas and fruit trees are more vulnerable to harmful insect larvae, particularly butterfly larvae. So A is correct, R is also correct, and reason clearly explains the assertion. So fourth option must be selected. Fourth one. Bacillus thuringiensis spores have inactive toxin which becomes active in the blood of harmful insect larvae that eat away the brassicas and fruit trees, causes disease insect larvae and kills them. Hence it is used as biological control to protect the crops from destruction of harmful insect larvae. Coming to the next one. Choose the incorrect combination. Ladybird controls aphids. Absolutely true. Dragonfly controls mosquitoes. True. The natural biocontrol agents, ladybird and dragonfly, they are natural. Trichoderma controls several root-borne plant pathogens. This is also true. Nucleopolyhedrovirus, bacteriophages. No. Nucleopolyhedrovirus. Right? It is a virus that selectively kills the insects. It is not a bacteriophage. Bacteriophage is a bacterial virus. But nucleopolyhedrovirus is a virus, but not parasitic on bacteria. It is parasitic on some selected insects. It is a baculovirus. Fourth option must be the incorrect combination. Let us see the answer. Fourth one. Nucleopolyhedrovirus is baculovirus that attacks insects. It is parasitic on selected insect pests. It is not bacterial virus. Matching again. Photoautotrophic biofertilizers means photosynthetic biofertilizers are blue-green algae or cyanobacteria. Among the four that is given list 2, Oscillatoria is a cyanobacterium. So these cyanobacteria are used as biofertilizers in particularly in paddy fields. So increase the nitrogen content of the soil, organic matter of the soil. They fix the atmospheric nitrogen into ammonia, but Oscillatoria is non-heterocystous cyanobacterium, Oscillatoria. Mycorrhizae forming biofertilizer, that is glomus. Glomus is the fungus that forms the mycorrhizae with the roots of higher plants. So B3, A4, B3. See, used as biofertilizer in legume crops, rhizobium. So rhizobium forms root nodules in legumes. Hence, rhizobium must be used as biofertilizer in legume crops. So C2. Free-living bacterial biofertilizer, azotobacter. Azotobacter is not symbiont, it is a free-living bacterial biofertilizer, D1. So, first option must be the correct one. A4, B3, C2, D1. First option must be the correct one. Photoautotrophic biofertilizers, Oscillatoria. Mycorrhizae forming biofertilizer, glomus. Used as biofertilizer in legume crops, rhizobium. Free-living bacterial biofertilizer, azotobacter. Coming to the next one. Which gas is mainly produced in fermentation of dough, production of beverages, and cheese making? Which gas is produced mainly? Carbon dioxide is produced. Carbon dioxide, not methane, not hydrogen sulfide, not hydrogen. Carbon dioxide, 19.1. So should be the one should be the answer. Let us see the answer. One. Carbon dioxide is mainly produced in fermentation of dough, production of beverages, and cheese making. Methane is mainly produced in biogas or gobar gas production. Hydrogen sulfide and hydrogen are subsidiary gases produced in anaerobic sludge digester of sewage treatment plants and gobar gas plants. 20th question. So identify the correct statements. Construction of STPs under Ganga Action Plan and Amana Action Plan was initiated by Ministry of Environment and Forest. True. Essentially correct. The objective of treating the effluent of primary treatment plant of STP in aeration tank is to decrease the BOD of effluent. This is wrong. Primary treatment plant of STP. No. It is secondary treatment, the BOD is decreased. So B is the incorrect statement. C. Nucleopolyhedroviruses are used as narrow spectrum bioinsecticides. Absolutely true. So that is why they are used in IPM, Integrated Pest Management Program. So A is correct, C is correct. Blue green algae as biofertilizers add organic matter to the soil. Absolutely true. Blue green algae not only improve the nitrogen content of the soil by fixing the atmospheric nitrogen, but also add organic matter to the soil, hence the soil fertility is increased. B is correct. B is correct. C is correct. D is also correct, but B is not correct. Mycorrhizal fungi impart resistance to root-borne pathogens of crop plants. Absolutely true. So the fungus of the mycorrhizae also protect the root from the attack by the root-borne pathogens. So B is incorrect, remaining are correct. So A, C, D, E. A, C, D, E. A, C, D and E. Third option must be the correct one. Let us see the answer. Is A, C, D, E. Should be correct. A, C, A, B, C, D, E. So primary treatment plant of STP in aeration tank is to decrease the BOD of effluent. So B is also correct. In aeration tank, the BOD is decreased. That is correct. The objective of treating effluent of primary treatment plant of STP in aeration tank is to decrease the BOD of effluent. So this is also correct. A, B, C, D, E. All the 4 are correct. All the 5 are correct. The aeration tank he has given, 4. Construction of STPs under Ganga Action Plan and Amana Action Plan was initiated by Ministry of Environment and Forests. The objective of treating the effluent of primary treatment plant of STP in aeration tank is to decrease the BOD of effluent. Nucleopolyhedrovirus are used as narrow spectrum bioinsecticides. Blue green algae as biofertilizers add organic matter to the soil. Mycorrhizal fungi impart resistance to root-borne pathogens of crop plants. That's all about the questions. You are requested to like, comment, subscribe and share.